So this is the case study about the broken dog by Navneet Plaha and Darius DeWitt. And here's our introduction. Extracellular, cellular, and intracellular signals. There are three kinds of surface cell receptors. The first is the enzyme-linked receptor, and they are found in all living species. The two important domains are, the first is an extracellular domain, which binds to a signaling molecule, and the intracellular domain has catalytic functions. The second kind of surface cell receptor is the G-protein coupled receptor, and that's found in the cells of all eukaryotes. The third surface cell receptor is the ligand-gated ion channel. So for question 1, we figured that ginseng must produce more nitric oxide. So this is because if more NO is produced, then relaxation is a lot greater, and therefore a much longer erection is produced. The second question, we noticed that the phosphodiester would get inhibited by the Viagra. So phosphodiester is responsible for breaking down cGMP and ending the erection. And if phosphodiester is inhibited, then the cGMP will not break down, and therefore PKG remains active. The phosphoproteins still move calcium into the smooth ER, and they also desensitize the effects of calcium, and basically the erection would continue. So for question 3, if both the chemicals aren't working, then we predict that there's a problem with the PKG or the P-lambin or the MLC protease. And if the PKG is defective, then it cannot signal the phosphoproteins to actively transport the calcium into the smooth ER. Therefore, the erection would never even start. And if the P-lambin can't work, then even if the PKG is telling it what to do, then it still wouldn't be able to pump the calcium out, so still nothing would be able to happen. For the MLC protease, and if that is defective, then MLC will never even be made, and MLC is what starts the relaxation, and so if that isn't made, then relaxation will never be able to occur. So the 5 things that PKG affects are the IP3, and so that basically inhibits that to release the calcium. The P-lambin, which also then releases the calcium. It also affects the MLC protease, which goes for the MLC and then relaxes. And it also affects the potassium being pumped out over here, as you can see. And it also affects the calcium directly, as you can see over here in the pump. So phospholambin is activated by PKG. When it is unphosphorylated, it inhibits the calcium pump, and this prevents the calcium from being pumped into the smoothie R, which causes the muscle to relax. However, when phospholamban is phosphorylated, it cannot inhibit the calcium pumps. And this is important because now that it can be pumped into the smoothie R, the muscle now contracts, and that just leads to the end of the cycle. Or not cycle, but of the process. PKG also inhibits, PKG now inhibits IP3. IP3 causes calcium gates to open, and it lets the calcium out into the cell from the smoothie R. And this causes the muscle cell to contract. And when inhibited, IP3 cannot open the calcium gates into the smoothie R, and this allows the muscle to relax. MLC phosphatase is activated by PKG as well, and MLC phosphatase, or the myosin light chain phosphatase, is the opposite of MLCK, or the myosin light chain kinase. MLC contracts when it's phosphorylated, to induce relaxation. MLC phosphatase inhibits MLC from being phosphorylated. This is unlike MLCK, which promotes the phosphorylation of MLC to make the muscle contract. Calcium and potassium. Calcium ion is inhibited by PKG from entering the cell, and this is because the calcium causes the cell to contract, and when you're looking to get an erection, you want it to be relaxed. And on the same level, potassium ion is activated by the cell, and this is because potassium ion close the calcium channels that let the calcium flow inside the cell. So if calcium is activated, if potassium is activated, then calcium cannot be let in, pretty much. And that is all for the PowerPoint presentation.